So, hi everybody. For those of you who don't know me, I have a PhD in computer science from the heyday of artificial intelligence, the second heyday in the 1980s, and I trained in pediatric endocrinology. And let's go right into it. This is a case history about the phenotype-first approach versus the genotype-first approach, and how one can balance it using autism as a case history. So modern medical science started with phenotype-first studies. And since we're talking about autism, one of the most common causes of autism is Fragile X syndrome, where kids have profound intellectual disability, large testicles, and a high frequency of autism. And it was presented clinically, phenotypically, and it was only by then doing an extensive family tree that the investigators found that it was X-linked. So this was a publication in 1943. And then 50 years later, the FMR1 gene on the X chromosome that is associated with Fragile X syndrome was identified in a sequence. So until the millennium, phenotype-first studies were the norm. Then what happened was with whole genome sequence or whole exome sequencing, we could now study entire exomes, so the 1.5% of the genome, which is codes for protein. And we could basically go through these patients, and we could compare kids with autism versus kids who don't have autism. And they were able to find literally 70 copy number variants, 70 small sections of the genome, where there were anywhere from a few bases to hundreds of bases that were either duplicated or deleted. And very significant, not present in the control population. And even though, so two things. First of all, amazing result, no phenotyping involved. And so a real validation of the sort of genetic triumphalist view of biology. However, if you talk to them, they'd actually be really annoyed with my characterizations. They'll say, Zach, we actually had a lot of phenotypes. And they did. They actually spent a lot of money through the Simons, Jim Simons is hedge fund, former professor at MIT, hedge fund billionaire, who I think has a child with autism and funded a lot of work. And they actually got a lot of phenotypes done by examination by expert therapists and cognitive psychologists, and these phenotypings, which are basically behavioral, extended the cost of this by a couple $1,000s, but they did not use the phenotypes at all. And just as importantly, the phenotypes were very narrow, focused on behavior. They weren't looking at the entirety of the healthcare of these children. Okay, so background: phenotypes first, millennium, advent of genetic triumphalism, genetics first. And then there was, after that study, there was literally dozens of studies identifying more and more loci that had representation in autism that did not have representation in so-called neurotypical individuals. So I then, for a variety of reasons that I don't want to get into, did a study, gene expression study of the blood. So gene expression is not genetic sequence, it's how much, okay, I'm letting in Anya Trail into the waiting room. I took care of it, Wendy. And I don't want you to know the details of it, except because it leads up to the next slide. So I did a study where I showed with reasonable accuracy that we could identify, just based on patterns of genes being turned on and off across 10,000 genes, whether a child was neurotypical or had autism. Turns out probably not good enough for clinical care, but interesting nonetheless. Because of this research, I was invited to Harvard Law School, where I saw this guy. And I knew as I showed up in this small conference room with a bunch of Harvard law professors and this guy that I recognized him from CNN. I couldn't figure out why. And then it became quite apparent when he said to me, Zak, why are you trying to commit genocide on my people? Now, those are loaded words for me because, in fact, I did lose some family members in the Holocaust. Plus, this guy happens to be Jewish Israeli. And so what did he mean by that? He meant that because I was developed a test that potentially could be used to screen for autism, we were going to eliminate people of his kind, people with autism. And he was saying, autism is just a different way of being, and we should respect that. I won't tell you what I actually told him very directly because this is family hour. Instead, I will tell you what a parent of a child with autism said, and you can Google this on the web. I actually didn't get this. Anya wants to get back in. I'm letting her in again. Okay, the mother said, I think that when Ari talks about autism and I talk about autism, we're talking about people with different clusters of autism. I know he doesn't like the word cure. If my daughter could function the way Ari could, I would consider her cured, said Singer. I have to believe my daughter doesn't want to be spending time peeling skin off her arm. And I want you to consider, for those of you who are clinicians, for those of you who are medical informaticians, for those of you who are geneticists, that it's bizarre as heck that we have a diagnosis so capacious so that having autism can allow you to be fully functioning TV personality and someone who is unable to speak and just picking their skin. It's a massive failure of naming. And I won't go into a longer pitch about why there are a number of economic and other forces which are keeping that diagnosis so capacious, because it serves several purposes, sometimes of pharma, sometimes of patients. And I can show you a lot of dollars, but it's very, very problematic. So I had done a study with a network of electronic health records systems across the United States looking at autism. And we found that there was a large, as has been suggested by literature, but this was now with 20, 30,000 patients, we saw an amazing overrepresentation of bowel problems in these children. 12%, I think, bowel problems, and a very large proportion of these with frank inflammatory bowel disease, where your gut is weeping blood, and it's not just a bad disease. And when I presented this to a noted behavioral pediatrician, he told me, and he said it this way, and he's otherwise a very nice guy, but it's amazingly dismissive. He said, quote, brain bad, tummy hurt. And you can imagine that similar things were told to many mothers and fathers of kids with autism, who the kid would be holding their abdomen and complaining of pain. And so this woman on the right, Finale Doshi, who's now a professor of computer science at the School of Engineering and Applied Science at Harvard University, had just gotten her PhD at MIT and was doing a 2-year postdoc with me. And I told her about this, and I suggested that she look in detail at the comorbidities, not the behavioral variation, but the actual medical comorbidities, the simple medical diagnoses of these patients. And looking across the 6,000 odd diagnoses that are scattered across these patients, and it's a power law, so that most patients just have a few of them, and a few patients have a lot of them. And what she did was quite ingenious. She turned each patient here into a row and represented each diagnosis, whether you had or did not have that diagnosis in a 6-month period, with a 1 or a 0. So if you had inflammatory bowel disease in this second 6 month, you get a 1 here. If you didn't have schizophrenia in the second 6 months, you get a 0 here, and so on. And so each 6-month block would have 5,000 bits describing that 6-month block comorbidities. And then what you do is simple clustering. So clustering, so making like patients come together. So patients having similar comorbidities at the same developmental time. That's all we were doing. And it was remarkably effective across this network of hospitals in sussing out the following. A group of kids going from here, 0 to 15, and this is the prevalence of epilepsy, so a group largely epilepsy. Another group, mostly immunological problems like infections, sinusitis, not shown here because it's lower frequency, also autoimmune diseases like type 1 diabetes, rheumatoid arthritis. And then a third subgroup, a lot of psychiatric disorders, from major depression to anxiety, not shown here because, again, very significant, but at a lower prevalence, schizophrenia. So we have now 3 pretty distinct patterns of autism across this population, and we haven't done any genetic studies yet at this point. And I will tell you that there were many who were still skeptical of our study, including the gastroenterologists. And so we went in and looked at multiple databases, a big study population with about 50 million people from Aetna. We looked at Boston Children's Hospital, and we looked at Wake Forest Children's Hospital. And we were able to see that compared to controls, kids with autism had far higher and much more significant prevalence of inflammatory bowel disease. And I want you to think about, just for a moment, what this means about this diagnosis. So let me explain. So I told you that I was trained as a pediatric endocrinologist, and I thought I was a pretty good doctor at the time, but never in my whole practice did I ever have mothers coming to me, as they did after we published the study, in tears of gratitude, acknowledging that their child did not have some random bad luck and unconnected disease, but this was part of their syndrome. And it's particularly bad because if you're not looking for it, what happens with kids with autism who don't speak? And the majority, unfortunately, have communication disorders, severe communication disorders. So if your 5-year-old child has abdominal pain, they say, "My tummy hurts, Mom." If a kid with autism who can't speak has abdominal pain, they act out. They go nuts. They're very difficult to handle because they're in pain and they can't say it, and they're like, moving around. So what do? As clever doctors, we give them major tranquilizers, antipsychotics. Note that antipsychotic does not take away from the pain. All it does is stop them from being able to complain about it. It's crazy stuff. And yet, that's what we've been doing. And this was the first time to show that we should be expecting a subset of these patients to have this constellation. Not at all obvious until, as informaticians, looking at the big picture. And by the way, lest you be over-impressed by the predictive abilities of genetics, and remember, most genetic variants are actually very weakly predictive individually. Here is a study that we use with that Aetna database, this insurance database, and we had huge numbers, as you'd expect for a disease that has prevalence of around 1%, of families with 2 kids, with 1 kid, 2 kids, and no kids with autism. And we could actually come up with a predictive model about the likelihood of having a second child with autism if your first child had autism, and with very, very tight error bars. So in fact, if the older sibling had autism and was a girl, and then you had a boy, there was 1 chance in 6 that the boy would have autism. Whereas because women are less susceptible to autism for a number of interesting reasons, if the first child with autism is a male and the second child is female, the female child has less than 1 chance in 20 of getting autism. And so these are very tight error bars, and we're looking at nothing except billing data. And so what I'm telling you is looking at the phenotype and taking it seriously is incredibly informative. This is work done by my then postdoc, he's now a faculty member, looking at the disease Tuberous sclerosis, which is known to be highly enriched for autism. It's medium rare. It's about 1 in 5,000. It results from genes mutations in 2 genes, and it goes all the way from being fatal with malignancies to just these weird tubers on your skin. And there's a lot of known conditions associated with tuberous sclerosis. So again, just looking at these claims databases, these insurance databases, we were able to look at 3,000 such patients. So 3,000 is actually a big cohort for this rare disease, but we expended 0 effort getting it because people have to pay for their care, or the state pays for their care. And so, and we had 1.5 million age and gender matched patients. So we one case of 500 controls. And so we identified all the codes, like instead of doing a genome-wide association study, we did a phenome-wide association studies, find all the phenotypes that had significantly different prevalences between tuberous sclerosis and non-tuberous sclerosis groups. And of course, because we're testing multiple hypotheses, we have to correct for multiple hypothesis testing. And so what you see here is the Manhattan plot that you obviously see for genes, where at the bottom is chromosomes, and here's the log odds ratio, the log p ratio, negative log p ratio of the significance of that genetic variant. But here it's negative log p-value of the clinical manifestation being different between tuberous sclerosis and the controls. And so there was quite a bunch of significant such findings. Many of them are cancers. Some are developmental disorders like autism and kidney cysts. So we actually recovered known, very well-known phenomena right with one click, essentially. But we found some new things like glaucoma, ulcerative colitis, and we also glaucoma and ulcerative colitis, which were actually at a lower rate than these other diseases. And for some reason, these patients are protected against cancer of the they were not protected. They had increased cancer of the brain and benign neoplasms of the lungs and the kidneys, but they were protected from bone cancer, previously not known. But the data is very, very clear, very significant odds ratio. And also, they are protected against these diseases: polycythemia vera, high hematocrit, Hodgkin disease, and myeloid leukemia. And why is that? It's because there's these genes, TSC2, is at the center of the mTOR pathway, which regulates a whole bunch of developmental programs, including and disorders of it are known to cause kidney problems, are known to cause cancers. And so what we're seeing is the full manifestation of that disorder that was previously not known by virtue of looking much more broadly. Okay, I'll skip by this slide so I can get it, because I'm making this talk a little bit more rapid than possible because of my unintentional tardiness, so that you can actually ask a few questions. So I then went on to ask with one of my former students. Now she's a assistant professor at the University of Beersheba in Israel. And we asked the question, can precision medicine allow us to go deep into the ASD subtype? And here what we were going to do was take very seriously what we call precision medicine. And I talk about precision medicine because I have a conflict of interest. The more people talk about money being conflict of interest, I think much more important is ideological perspective. So my ideological perspective is around precision medicine, because I was on the National Academy of Science committee that helped write the report. And basically what we said was, we envy Google Maps because they're able to layer all these different data types and add so much value onto geography. And we'd like to be able to do the same for all the different clinical measurements and genomics and proteomics and environmental studies, stack them all together instead of the common coordinate system being longitude and latitude, the common coordinate system would be the individual. So can we do this to better understand autism? So we use the following data types: whole exome variation, so mutations in looking at the coding genome. We looked at segregation patterns, looking at how these traits move from one part of the family to the other. We looked at expression correlations, that is, we looked at how genes were being turned on and off, as you'll see in the brain. And we focused particularly on genes that had different expression patterns, turned on and off differently with different trajectories in males than females, because again, as I hinted to you before, males have much more fragility relative to autism than females, and the pretty reproducible number is there's a 4-to-1 ratio male to female of autism. And then we use electronic medical records to validate our study. And note that, and health claims data. So note that we're using two different types of phenotypes: molecular phenotypes and clinical phenotypes, to triangulate with the genotype to find groups in the middle, to find the subsets of patients occupying a subspace of this precision medicine multimodal space. I just want to drop here a plug for the fastest accepted paper I ever wrote. Actually, I didn't write it. My friend Griffin Weber wrote it, but I told him to make a diagram showing how all the different data types that we need to have to do precision medicine come together, and included tweets and medications and memberships and gyms and so on. And so we sent it to the Journal of the American Medical Association. And within 6 hours, we're told it's accepted. And it was only because of this paper, of this diagram. And because it's a very evocative diagram. In fact, I've been running into people who have this diagram on their lab wall because it shows what the universe is. And so this is just my way of saying the study I'm about to describe to you takes this approach quite seriously. So if we look at the whole exome, every one of us has hundreds of thousands of variants, or tens of thousands of variants. Which one are we to look at? Some are common, some are less common. And how do we integrate this with the fact that you can have a variant which may or may not express itself in different families, but there's clearly something going on, because you can have kids in the same family that don't manifest the same thing. And so you presume, you hope that you can track the correlation between the trait and the variant. Okay. And because this is a developmental disease, we believed in doing this study, the expression of interest, the expression pattern of genes of interest was in the developing human brain. So that, since it's a developmental cognitive behavioral phenotype. So we need to be able to look at the gene expression in the brain. All right, and so I already told you about this. So it so happens the world has accumulated a lot of very interesting databases, and those of us who are fortunate enough to be biomedical informaticians are very lucky to have all these data sets that are available. So, for example, BrainSpan includes 524 human samples, humans with samples at multiple developmental stages, all the way through gestation. I think from 12 weeks, all the way through to 40 years, and in looking at different parts of the brain, whole exome sequencing. And so Alal Aran, this postdoc, now a faculty member in Ben Gurion, said, let's look at all the gene expression patterns with development and find the ones that are most different between males and females. And we found a bunch of such patterns. And so that gave us a list of genes that were most sexually dimorphic during development. We then looked at the several studies of autism, and from the NIH database obtained exomes, and we said, basically, of all the rare variants that we identify in these exomes, what is happening with the variants in those genes, only in those genes, the small set of genes that we found to be massively discordant between males and females. And so looking at these multiplex families and these discordant pairs, we're able to find the subset of those genes which had mutations, rare mutations, affecting function. And what we found was there were a few clusters of such genes. And these clusters were immune function, transcriptional regulation, synaptic function, and growth and morphogenesis, development of organs. Those first four had been published somewhat in the literature. The lipid regulation was a new cluster. And so we found that pretty interesting. And so we asked ourselves, well, what can we now do to show ourselves that this is actually something truly relevant to patients in the real world? Well, we said, where are these mutations happening? And it turns out, these mutations are happening in this pathway. And this pathway involves this lipolytic process to release free fatty amino acids. It involves LDL particles binding to LDLR. So mutations in any part of this could result in a disorder here. And so we focused on this pathway, which, as you see, is highly significantly mutated compared to the neurotypical patients. And so we now have a hypothesis to test that lipid dysregulation might be a convergent etiology in autism. Yes, for example, like there. And so we did a study of blood profiles in children with ASD. And sure enough, we found that both LDL, total cholesterol, triglycerides were all elevated significantly. You can see the confidence intervals, and you can see the p-values below of this in our patient population. Okay, then we look back at the Aetna database, which is much larger, again, on the order of 50, 60 million. And there again, we saw a very significant enrichment for hyperlipidemia, mixed hyperlipidemia, and hypercholesterolemia, and so on. Again, highly significant. And then we found that the parents actually had some, but nowhere near as much dyslipidemia or as severe dyslipidemia as the kids. So the mothers and fathers had some, but again, nowhere near the odds ratios that the children did. So could this all be perhaps just that families are eating poorly? And if mom and dad eats poorly, it's very likely the child eats poorly. So how did we address that? So we went into the 23,800 families, almost 24,000 families, where one child was diagnosed with autism and one child was lacking such a diagnosis. And so sure enough, same family, but if the child had the diagnosis, the odds ratio was over 1.5, which is a big signal and very significant in the affected versus the unaffected controls. Then when we looked at mouse models where others at the Jackson lab had knocked out all the genes one by one, not only the genes in this pathway, but all genes, but if you look at the genes that are related to dyslipidemia, many of the reported phenotypes of these mice are more autistic, than some of the classic autistic models of mice. And the way to think about it is we should not be surprised about this in retrospect, because after all, lipids are not just a source of energy, but they are incredibly important in human neurodevelopment. Depending on who you talk to, they're either 50 to 60% of dry brain weight. And they're the building blocks of cell membranes, neurons, astrocytes, myelin sheaths, crucial for maintaining neuronal excitability and synaptic function. And we're beginning to understand, by the way, that in neurodegeneration, for example, slight defects of that will cause premature Alzheimer's. So the ApoE gene codes for a protein that's absolutely important for trafficking cholesterol from astrocytes to neurons. And slight mutation of that puts you at a much bigger increase risk. And these fats, these lipids, have multiple roles in lipid signaling and immune mediators. And so this starts to beg the question, could maternal diet during gestation actually affect this? And so it's an unanswered question, but recently, independently, someone just published a study, like within the last month, that mothers who have type 2 diabetes uncontrolled during pregnancy or poorly controlled have a much higher rate of kids with neuropsychiatric disorders. So in summary, a genetics first approach, although initially extremely successful, risks scientific myopia for understanding complex disease. It's very much like saying, we're going to do genome-wide association studies on heart failure. And we're going to put all the patients who have heart failure into one bucket and all the ones who don't have heart failure in the other bucket. We will not distinguish between the heart failures that are due to valvular problems, that are due to atherosclerosis, that are due to viral infections, or that are due to protein disorders. But that's exactly what we found here in autism, and that's what's almost certainly true for many other disorders like inflammatory bowel disease and so on, that we're mashing together all these different diseases into one disease, and we're not using the phenotypes just as carefully as we would use the genotypes to tease apart these diseases. And so a phenotype first approach is very helpful in this big picture, but it's missing the mechanistic insights. It tells you that there are more diseases here, and they're worth studying individually, and they're worth treating individually, but don't tell you exactly what's going wrong. And so I would say that if there was a moral of the story here, it's that we need a combination of phenotypic and genetics approach at the same time to meet in the middle, not to do one first, because either you get myopia or you get wide angle without understanding what's going on. But this is a problem because we see across multiple academic centers that the people who are doing clinical phenotyping are not the same people as who are doing genomics. So there's a language collaborative mismatch. And the data acquisition that you need for one, electronic health records, reimbursement codes, are very different kinds of data that have their own weirdness, that have their own noise, that have their own biases, than those biases and noise that you see in genomic data. And so it really requires significant multidisciplinary skill and multimodal data access, which I think is a challenge for all of us in medicine, and particularly those of us who are in biomedical informatics, to take on front and center.